Thank you all for being here today. I'm truly delighted to speak to the UPNA community, and I also wanted to thank the Cognitive and Behavioral Neuroscience Group and Organization Committee in particular. Thanks to Pablo Risotto and Raul Cacho for the invitation to share my research in this space. I am an assistant professor at the California State University here in Monterey Bay. And while most of the research that I am going to present today uses mouse models, I'm also currently translating some of this research into studies with humans and fish models in collaboration with various groups. So now with this presentation, I'm going to share with you some of our research on the mechanisms by which the peripheral systemic response to physical exercise affects the brain. So it is commonly accepted that physical activity improves overall health in many organs, and this includes the brain. So several studies show that exercise is beneficial for cognitive function during aging, including healthy aging, brain damage, and neurodegeneration, and that it can reduce the risk for dementia. So studies with animals have linked some of these positive effects to the hippocampus, which is an area of the brain that is essential for learning and memory. So in particular, what these studies show is that physical activity increases brain plasticity, which is the ability of the brain to grow and reorganize. So these changes can be in the form of increased neurogenesis, which is the creation of new neurons, increased angiogenesis, which is the creation of new blood vessels, and enhanced synaptic plasticity. In addition to neuroplasticity, the positive effects of exercise on the brain have also been linked to decreased neuroinflammation in the hippocampus. Now, how does exercise induce these effects on the brain is still unknown. So one possible explanation to how exercise affects the brain is the idea that the brain responds to circulatory factors that are released from peripheral organs and tissues in response to exercise. So if we think about the act of exercise, this is a voluntary movement that starts on the brain and signals travel down the spinal cord all the way to the muscles to generate the movement. But at the same time, in parallel, neural signals are also going to stimulate the cardiovascular, the respiratory, the metabolic, and hormonal systems to keep up with the demands of the movement and keep the body within homeostatic conditions. The activation of these systems results in the secretion of different factors in the circulatory system, as you can see here. And the circulatory system allows for these factors to target different tissues in the body, such as the brain. So the hypothesis here is that the effects of exercise on the brain could be mediated by circulatory factors that are secreted peripherally by these organs and tissues. And so supporting this idea, a growing number of studies show that physical exercise increases levels of many proteins in plasma, which also have been shown to increase hippocampal neurogenesis in animal models. And this includes IGF, which is insulin-like growth factor 1, vascular endothelial growth factor, which is VEGF, and irisin. So, however, it is unknown whether exercise conditioned plasma that contains the proteins induced by exercise at physiological levels is sufficient to benefit the young, healthy brain. And what are the overall effects of exercise conditioned plasma on the brain? So in order to study the effects of exercise conditioned plasma on the brain, we use a simple model of plasma transfer from mice that exercise in a running wheel for 28 days into sedentary or non-exercise recipient mice. And our control group received plasma from mice that did not have access to a functional running wheel. So we administer 8 doses of plasma over a 28-day period to match the physical activity regimen from the donors, and we use the retroorbital vein to deliver the plasma. Then we study the efficacy of exercise plasma to alter hippocampal neuroplasticity and to improve performance in hippocampal-dependent behavioral tests. And both of these endpoints are widely recognized to be improved in mice with exercise, with physical exercise. So using immunohistochemistry and markers of cell proliferation in the dendritic gyrus of the hippocampus, we observed that sedentary mice that receive treatment with runner plasma show an increased number of neuroblasts and neural stem progenitor cells. As shown here in this micrograph, mice that received runner plasma had more DCX positive cells, which is a marker for neuronal precursor cells or immature neurons. So using BRDU and EDU as markers of cell proliferation, we also observe increased number of proliferating and surviving cells at a 28-day time point. And so we identified that some of these cells were neurons and astrocytes. So these results suggest that some of the benefits of exercise on neurogenesis can be transferred by transferring plasma from exercising mice to non-exercise recipient mice, and that the physiological levels of plasma factors are sufficient to induce these effects. So we also wanted to determine whether runner plasma could have any impact in contextual memory, which is an hippocampal-dependent type of memory. So to do this, we used the fear conditioning test. And in this test, mice learn to associate a preceding tone and a context, which is the case, with a fearful stimulus, in this case, a mild electrical shock. So once the association is formed, we can then test the memory of this association by measuring freezing behavior in response to the tone, which we call cute memory, or in response to the context, which we call contextual memory. So we observe no significant differences between mice treated with control plasma or runner plasma in the cute memory test, but we observe increased freezing behavior in mice that receive runner plasma in the contextual memory test, suggesting that runner plasma improves contextual memory of a fearful stimulus. We also studied spatial memory with the water maze test, which is another behavioral test that is associated with hippocampal function. And with this test, mice are trained to locate a platform hidden in a pool under the water. And let's see, I lost my mouse here. Here is the platform. And so you train the mice, and here what you can see is that at the beginning, the mice take a long time to find the platform, and you see a cap here in 60 seconds because we don't let them swim more in the water at the beginning, only 60 seconds. But with the trials, they learn that the platform is hiding there. They find it very quickly, like within 20 seconds. So then after the training, if you remove the platform, you can then analyze their behavior around the target area, which is indicative of their spatial memory. So we found that mice that receive runner plasma spend more time in the virtual platform and around the area, which supports our previous findings that runner plasma improves performance in hippocampal-dependent behavioral tests. One interesting idea that we have heard about and we have explored as well is that that could explain the positive, something that could explain the positive effects of runner plasma on behavior is the possibility that runner plasma, but no control plasma, could increase the overall activity levels on mice. As animals with runner plasma will be more active. So in order to explore this possibility, we analyzed the behavior of animals treated with runner plasma or control plasma in the activity chamber, and we observed that control plasma and runner plasma infused mice show very similar activity and velocity patterns, as you can see here. And we also analyzed the preference for light, dark area, which resulted in similar time spent in the light area for both groups. Additionally, we also analyzed the distance travel, tachypnotaxis, and velocity displayed by mice infused with runner plasma and control plasma in the water maze and found no differences. So these results suggest that runner plasma doesn't alter overall activity levels in mice and support our previous findings that runner plasma improves performance in hippocampal-dependent behavioral tests. And at this point, what we wanted to know is what is doing runner plasma in the hippocampus at the molecular level. And to do this, we analyzed the hippocampal transcriptome of mice that received runner plasma and control plasma by using RNA sequencing in the whole hippocampus. we observe 1,974 differentially expressed genes, or DEGs, of which 61%, I lost my mouse again, 61% were downregulated and 39% were upregulated. So we observe downregulation of genes implicated in cell migration, such as TNS4, cell adhesion, such as IGSF5, and upregulation of genes implicated in hippocampal synaptic function during learning and memory formation, such as FOXO6, and immune system and plasticity, such as FAM19A2. So functional analysis of the top genes by gene ontology and summarized into non-redundant hierarchical terms by Rebigo pointed to a downregulation of acute inflammatory response as the most significantly affected biological pathway, here the circles in yellow, orange, followed by upregulation of cell junction organization and negative regulation of proteolysis. So these findings indicate that factors in plasma from running mice are sufficient to downregulate the baseline expression of inflammatory genes in the young adult hippocampus. And so we then wanted to know whether runner plasma could indeed have an impact on neuroinflammation. To test these potential anti-inflammatory properties of runner plasma in the brain, we use LPS as a model of acute neuroinflammation. So mice were first inoculated with LPS and then treated for a period of 8 hours with control plasma or runner plasma or saline for both time points. And we perform RNA sequencing in the hippocampi of these 4 groups. So we have saline, LPS, saline, LPS, control plasma, and LPS runner plasma. So we observe 4,936 differentially expressed genes in the hippocampus in response to LPS. We found 1,082 DEGs in response to runner plasma during inflammation. And of these, 693 genes were significantly changed with LPS and with runner plasma. So here, these 693 genes are plotted in a double volcano plot in a way that the x-axis in this graph represents the p-value and the direction of the effects of LPS. And the y-axis represents the p-value and the direction of the effect of runner plasma with LPS over control plasma with LPS. So what we observed was that treatment with runner plasma but no control plasma reverse the expression of a large number of these genes induced by LPS. So all of the genes that were upregulated with LPS are on the right side of this axis. And as you can see, those are the genes that where many of these genes were downregulated with the treatment of runner plasma. So this relation resulted in a negative correlation on these proteins. We also use PCA analysis for visualization purposes. And what we observe is that the transcriptome, the hippocampal transcriptome of runner plasma treated mice had a greater similarity to the control mice that did not receive LPS. So you can see here the green group is our LPS treated group with runner plasma. So these results suggest that runner plasma is anti-inflammatory in the brain. So we validated some of the top genes using qPCR. Some of these genes belong to the toll-like receptor signaling pathways, such as MID88, TLR1 and 2, IRAK3, and others belong to the interferon pathways, such as IRF9, interferon gamma receptor 1, STAT1 and 3, and GFAP. And what you see is this pattern in which in the group of the control group that only receives saline, there's the blue group is the control that receives saline, this is our baseline. And then when you inject LPS, you see the upregulation in almost all of these genes, except for P2RY12, which what you observe is a downregulation. Then when we treat these animals with control plasma, we see that the differences are not significant. But when we treat it with runner plasma, we see that these changes induced by LPS are moved towards a more baseline type condition. So in this case, they are downregulated, bringing them closer to to our control group, saline group, except for P2RY12, where we see an upregulation. So until here, I have shown you that data that support the idea that exercise plasma increases neuroplasticity, improves the performance in hippocampal dependent behavioral tests, and decreases the expression of neuroinflammatory genes in mice. So what we wanted to know at this point was to understand what in plasma is mediated these effects. So to this end, we analyzed the proteomic profile of our runner plasma and control plasma samples using mass spectrometry. We used a shotgun approach, and then we validated it using TMT. We identified 235 proteins simultaneously detected in both groups. And of these, 49 proteins were significantly changed with running. So biological pathway analysis pointed to changes within the alternative and complement pathway. In fact, we observed that 26% of the proteins significantly changed with exercise belong to the complement and coagulation pathways. And we're looking at the proteins from the alternative complement pathway. We also observe an upregulation of two main inhibitors, clustering and factor H. And additionally, we also found an upregulation of C1 inhibitor, which is an inhibitor of the classical pathway. So changes in these proteins suggest that these pathways are inhibited with exercise. And later in the presentation, I'll show you some experiments with it to begin to test this hypothesis. But for now, what we wanted to know is whether these two main inhibitors of the alternative pathway can play a role in the anti-inflammatory effects of runner plasma in the brain. So to this end, we immune depleted runner plasma from clustering, from factor H, and for comparison, we also immune depleted PEDF, which is pigment epithelium-derived factor, and LIFR, which is leukemia inhibitory factor receptor. So, and both of these are known to be anti-inflammatory as well, and were among our top increased proteins. So we use magnetic beads coupled to an antibody to generate plasma from runners that were depleted from CLU, depleted from factor H, PEDF, or LIFR. And we use the resultant plasma to treat mice injected with LPS. And then we use qPCR to analyze the genes that were previously shown to be altered with plasma in the hippocampus. Here, we can see in blue is the baseline expression of these genes without LPS, and then we add LPS, the expression of these genes increases, except for P2RY12, which is downregulated. Now, in animals that receive runner treatment with runner plasma, we observe a reduction of expression on these genes, bringing them closer to baseline conditions. This is the same thing that I showed before. Now, what I am going to show you is the gene expression on the groups that receive treatment with either one of these four different types of plasma depletion. And what we observed was that depletion of clustering in runner plasma largely eliminated the anti-inflammatory properties of runner plasma. Depletion of factor H had a lesser effect on the anti-inflammatory capacity, and depletion of PEDF or LIFR had no or very little effect, as you can see here, indicating that plasma clustering is an important factor in reducing brain inflammation via exercise conditioned plasma. In addition to our findings implicating clustering as a mediator of the anti-inflammatory effects of runner plasma in the brain, we found that LRP8, which is the main receptor for clustering, is highly expressed in the brain. And that brain endothelial cells, here the second line, show by far one of the highest expression across the organism of this LRP8, which made us question whether brain endothelial cells may be the immediate target of circulatory clustering. So to answer this question, we injected recombinant clustering labeled with the fluorophore ATO647 or PBS with the fluorophore as a control. And then we analyzed the brains with confocal microscopy. And we observed a colocalization of lectin, which is a marker of the overall cerebrovasculature with the fluorophore, indicating that clustering is localized within the cerebrovasculature, as you can see here. To further validate these findings, we also use SMA, which is a marker for arteries and capillaries, and TFRC, a marker for veins. And we saw again that recombinant clustering binds to arteries and capillaries and veins, as you can see in these two micrographs. So these images suggest that clustering may be acting on the brain via the brain endothelial cells. So to answer this question, we did single-cell sequencing on hippocampal brain endothelial cells of mice with neuroinflammation. To this end, we repeated our LPS model, and we treated mice with recombinant clustering, and then used FACS sorting to isolate hippocampal brain endothelial cells and perform single-cell sequencing of these cells. For comparison, we also used the transgenic mice overexpressing mutated APP as a model of chronic neuroinflammation. And these Disney plots show the different populations of brain endothelial cell types that we use for our studies. And by comparing the results of these two studies, we aim to begin to determine an anti-inflammatory signature of effects of clustering on brain endothelial cells. So to this end, we combined the results, as I mentioned, from both experiments and found that clustering reduced the expression of 20 different immune genes that were upregulated with chronic and acute neuroinflammation. Pathway analysis of these 20 genes pointed to a downregulation of the interferon pathway, suggesting that the interferon pathway in brain endothelial cells play an important role in mediating the anti-inflammatory effects of clustering in the brain. I'm going to change gears a little bit here, but to begin to determine the validity of these findings into humans, we collaborated with Dr. Casey Fairchild, who is a clinical psychologist at the VA hospital in Palo Alto here in California. And we analyzed the plasma proteome from 20 veterans that suffer from mild cognitive impairment. And we did analyze the proteome before and after a 6-month physical exercise intervention in which participants exercised 3 times per week for 1 hour per day. And these sessions included 30 minutes of continued aerobic exercise, things like treadmill walking, stationary cycling, and 20 minutes of full body resistant exercises. So consistent with our findings in mice, we found that proteins from the complement pathway were also changed in patients after physical activity, including an upregulation of clustering and decreased levels of C3, which indicates that the complement pathway could be inhibited. Together, these findings show that exercise in humans and mice results in upregulation of clustering and downregulation of proteins on the complement system. So when directly comparing proteins of the complement and coagulation pathway induced by exercise in mice and humans, we see this concerted upregulation of inhibitors in red here and downregulation of proteins from these pathways, which suggests that the function of the coagulation and complement systems could be downregulated. So to begin to answer these questions, we use an ELISA kit to assess the formation of membrane attack complex in samples from runners and controls. However, we observed no differences on membrane attack complex formation in vitro when the classical, the alternative, or the lectin complement pathways were induced by AGM, LPS, or MBL. We also assessed fibrinolysis and measured clot formation over time and show a significant reduction in the maximum clot form in runner plasma compared with control plasma right here and here as well. But we observed no differences in clot lysis time or the time to half lysis was found in runner on control and samples from runners or control mice. So these data suggest that clotting cascade could be inhibited in runner mice. So to summarize, here we show that peripheral factors released in plasma in response to exercise are sufficient to recapitulate the beneficial effects of exercise on neurogenesis, learning and memory, and to reduce neuroinflammation in the hippocampus on non-exercise mice. Clustering plays a critical role mediating the anti-inflammatory effects of runner plasma in brain endothelial cells and the whole hippocampus, possibly via the interferon pathway. and physical activity in human patients with mild cognitive impairment, so similar upregulation of plasma clustering levels. And with this, I wanted to acknowledge the Tony Wyss-Coray laboratory at Stanford, the Elias lab, also at Stanford, that perform all the proteomics, the laboratory from Casey Fairchild at the VA hospital, my laboratory here at CSUMB, and to various sources of funding. And